Hello, my name is Lei Zhao from Taxell Therapeutics, and today I will introduce one of our novel AAV capsid with RGD-free sequence and has very strong cardiomyocyte and myocyte tropism, and also have very strong liver detargeting property. This is my disclaimer. Okay, a quick introduction for Taxell Therapeutics. We have one platform for development of AAV novel capsid by directed evolution and screening in non-human primates based on DNA shuffling library. And currently we have three novel AAV capsids. Number one, ID31, target to the heart and skeletal muscle and has very strong liver detarget effect property. And we deliver SERCA2A or FGF21 for the heart failure, and also we deliver insulin to the skeletal muscle and for T1D or T2D as well. And the second one is ID17503, which target to the liver, has very strong liver transduction efficiency, and we deliver FGF21 for MASH. And the third one is AV number 11, a target to the brain neurons. And we have a lot of a very open strategy for collaboration. We can do this out licensing based on this capsid, novel capsids, and also out licensing for the novel pipelines as well. Okay, so ID31 is selected from 2 runs from non-human primates. And after 2 rounds, we have a quick validation from the top 50 candidates. ID31 is performed the best here, so you can see in the heart and skeletal muscle, and both in the DNA and the RNA level. And we also co-incubate with the AAV9 and AAV-RH74 with different barcode. So you can see from the NGS sequencing, the ID31 performed very good in the biodistribution, both in the DNA and RNA level in heart and skeletal muscle, and much better than AAV9 and AAV-RH74. And in liver, ID31 is just located here, so that means the DNA is very low, has very low biodistribution in liver, and also very low mRNA level in the liver as well. And in another independent study, pool study, we just mixed ID31 with our benchmarker MyoAAV4A and 4E, which published in Cell in 2021, and also with the AAV9 and AAV-RH74. And we also separated different parts in heart, such as left and right ventricle and atria, and also different parts of skeletal muscle. And you can see the data for the mRNA level versus AAV9, so that you can see the ID31 is performed the best in heart and skeletal muscle, and also has the lowest level in the liver as well. So in the dose escalation study, ID31 is head-to-head study with AAV9 in 2 doses, 2E13 Vg per kilo and 2E12 Vg per kilo. And that is performed in the 2 monkeys. That you can see both in the high and the low dose, ID31 performed like 9-fold transcription level versus AAV9 in RNA level. And also in the skeletal muscle, it performed 20-folds versus AAV9. But in liver, it's reduced like 20-folds mRNA level in AAV9 in liver as well. And if we check the protein level that we just monitored by IHC, that ID31 performed very strong transduction in heart and skeletal muscle as well, but very low in liver. It's almost negative in liver. And we also quantified this in cardiomyocytes by pathologists. And this is the ID31 versus AAV9 in cardiomyocytes, and here in the hepatocytes, so it's pretty low in the liver as well. And when we want to check the cell specificity for this novel capsid, so we have the staining for the myoglobin in both in the heart and skeletal muscle to stain the cardiomyocytes or myocytes. And ID31 is delivered EGFP. So when we see the co-localization with this red and green color, and it shows like the 88% of the infection of ID31 was in cardiomyocytes or myocytes here. And we don't use any tissue-specific promoter. We use the CBA, that is a ubiquitous promoter. So that means the most of this cell specificity is come from this novel capsid, but it's not from the promoter. Okay, and to study the molecular mechanism for ID31, we use CRISPR screening technology and cryo-EM to study it. And by CRISPR screening, we identify several hit genes, and one of them is AVR, and the others are the enzymes for the glycan chains biosynthesis. And it refers to, the two major glycans. One is sialic acid, and the other is heparin. So by using these two assays, one is neuraminidase. We use neuraminidase to remove the sialic acid on the surface and expose the galactose. And you can see the different profile, the binding profile generating by ID31 is quite different from this wild-type AAV2 or AAV9, but quite similar with AAV1. And in the heparin block assay also showed that ID31 is not sensitive or has very low binding affinity with heparin. It's not like AAV2 or AAV9. And in the structure information that we can see a very unique binding pattern with ID31 and AVR. It has conformational change from the side chain of the serine. And also it shows like the decreased 10-folds reduced affinity with AVR compared with the wild-type AV as well. Okay, so when we have this very good tropism for ID31, we try to use it to replace AV1 and to improve the state-of-the-art clinical trial with the SACA2A delivery by the intracoronary delivery. So that we see that from the clinical data from the papers, SACA2A and I1C are promising targets for the heart failure, and the way we use is also just to replace AV1 with ID31. And in the tech model in mouse, we delivered both SACA2A and FGF21 by ID31. That you can see from the data after 1 month after the single IV infusion, the black bar is the tech model, so you can see the ejection fraction is very, very low here. But the dosing group, especially from the SACA2A group, can recover the phenotype pretty good. And also you can see from the imaging, like the enlarged abnormal heart in the tech group, but the dosing group, the SACA2A group, has very good and normal size of the heart, and also the area of the fibrosis is also improved. And we also try to do this head-to-head study with ID31 delivered SACA2A versus AV1 delivered SACA2A. And we dose the non-human primates by intracoronary infusion. The dose is 1E12, which is comparable to the 1E13 VG in the human. And also we use the NHP and we use intracoronary. And you can see from the data and in different parts of the heart, especially in the ventricle, ID31 performed 3 times better mRNA level than AV1 delivery. But in liver, it reduced like 3-folds versus AV1 delivery. And the most interesting thing is ID31 has very good transcription efficiency than AV1. So we just calculated, it's just almost in heart almost 10-folds or 9-folds increasing for the mRNA generation compared with AV1 in heart. And we want to find the best way for the administration for ID31. So we use this study by using 4 different barcodes and two different administration. One is IC and one is IV. And we test it in the non-human primates. And from the data shows pretty interesting that the ID31 increased 3-folds mRNA level by IC infusion compared with AV1 IC. So this is quite similar, similar with the data from the SACA2A. And also in the IV data, it seems even better. Delivery by ID31 is increased 5-folds mRNA level in heart. And in liver, ID31 also shows pretty low level compared with AV1 in both IC or IV. And the interesting thing is the IC infusion seems like to help AV1 a lot in the very good proximity with LVA, so that is left ventricle anterior. But it's not pretty a lot with ID31. So that probably means ID31. The best way for ID31 is IV infusion, but not IC. And also, it has great potential for escape from human IVIG inhibition of ID31. We select from our donors 3 groups, group 2, 3, and 4. They have different level of inhibition neutralizing antibody with ID31 and AAV1, both ID31 and AAV1. And within each group, ID31's inhibition is much stronger than AAV1. And the group 1 is the negative control that we only use PBS but no human serum. And from the data that you can see, we co-incubate in the nude mice, and after 1 month, and then you can see both in the heart and skeletal muscle, the groups, group 2 and group 3, even especially group 2 seems like the same level with the negative control. That means even the neutralizing antibody seems like the positive for ID31, but it still remains very good potency, in vivo potency, that in fact with heart and skeletal muscle. But in liver, that you can see in the different group, the AAV1 shows much more MRA level than ID31 as well. So in summary, ID31 has very good heart and muscle targeting properties, like the 9-folds versus AV9 in heart and 20-folds versus AV9 in muscle, and also reduced 20-folds in liver. This is the data is generated from 2 different monkeys. And also it has very good potential by using the IV infusion instead of the IC infusion to replace AV1 SRCA2 as well. Okay. So if you're interested in our novel capsids and also our pipelines, just please contact me. Feel free to contact me by email or by my WeChat QR code. Thank you.